Welcome to this lecture on eukaryotic protein expression systems 2. In the previous lecture, we had discussed about the expression of proteins in yeast and insect cells, and before that we had discussed about introducing genes into E. coli cells and how we can make proteins in E. coli cells. And when your protein actually requires specific post-translational modifications such as glycosylation, phosphorylation or specific folding involving disulfide bonds, and in such cases many a times proteins of eukaryotic origin, when they are expressed in E. coli cells, you will not happen, and therefore the protein may not have proper biological activity. So in such cases you have no other option but to take your gene and express the eukaryotic expression systems under the control of a eukaryotic promoter in a eukaryotic cell line. And the first choice to express a protein of eukaryotic origin in a eukaryotic expression system is again microbial systems, and yeast is an ideal choice to express some of these mammalian proteins in yeast cells. In fact, a number of mammalian proteins have been expressed successfully in yeast cells and are now available as either diagnostics or therapeutics in the market. But many a times when you express the proteins in either insect or yeast cells, the protein may not be biological active because the folding or the glycosylation pattern or other post-translational modifications is not exactly same as that happens in a mammalian cell types. So in such a scenario you have no other choice but to go and clone your gene in a mammalian expression vector and express this in a mammalian expression system. So let us now discuss today as to see what kind of mammalian expression systems are available and how does one go about expressing a gene in a mammalian expression system. I am also going to spend some time to discuss about cell-free protein expression systems, which has a number of advantages as well as disadvantages over expressing genes in cells and cell lines. And in the next class we are going to talk about the expression of proteins in plants and farm animals, and then we will talk about human gene therapy, where instead of taking your gene and expressing in cells outside, you now are directly going to express your gene in the body itself and see whether you can correct some of the genetic disorders by using this kind of a human gene therapy approaches. So let us today focus on the expression of genes in mammalian cells and how we can make recombinant proteins using mammalian cells as factories. As I said, let us now spend some time to understand this logic. Why should you express a protein, or why should you produce a recombinant protein in mammalian cells? As I said, when you want your recombinant human protein or a recombinant mammalian protein to be exactly the same as this occurs in the native form, if you want to express human protein in a recombinant form, and if you want this recombinant human protein to be exactly the same as it is present in the native human cells, then you have no other option but to go for a mammalian expression system. The reason is, many a times the prokaryotic as well as yeast and insect expression systems may not be able to produce mammalian proteins in the same way as it happens in the mammalian cells, and when the post-translational modifications or the folding patterns in these recombinant systems are not exactly the same as that happens in mammalian cells, then the mammalian protein that is expressed in either yeast, insect or prokaryotic systems will not be the same as that happens in vivo, and therefore you have to choose a mammalian expression system for your recombinant protein production. One of the most important things that often happens is the proper disulfide bond formation. As you know, many proteins contain number of disulfide bonds present between the two cysteine residues, and this disulfide bond formation is catalyzed by the enzyme disulfide isomerase. And many a times the proper formation of these disulfide bonds is very important for the correct folding of the molecule. And when these disulfide bonds are not properly formed, then the protein that is produced in this recombinant form will not be biological active, and therefore it will be useless. So proper disulfide bond formation often happens only when you express these human proteins in a mammalian expression system rather than expressing in a yeast or insect systems. So you have no other option but to go for a mammalian expression system for making your recombinant protein. The other thing that normally one encounters when you want to make recombinant proteins is proteolytic cleavage of precursor forms. Many a times in human and other mammalian cells, the proteins are often proteolytically processed to get a final mature form, and when this kind of proteolytic processing is different in either insect or yeast cells, then you cannot make this protein in those systems. So you have to come back to the mammalian expression systems for making your protein. And the most important thing that often happens for you to choose a mammalian expression system rather than a yeast or insect expression system is the glycosylation pattern. Now, although the yeast and insect cells do glycosylate proteins, many a time the exact glycosylation that happens in mammalian cells may not be reproduced in the yeast or the insect cells. And if these glycosylation differences are going to affect the biological activity of protein, then you have no other choice but to express your protein in a mammalian expression system. And as you know, glycosylation primarily takes place on serine and threonine residues when it is actually called as a O-linked glycosylation, and when this glycosylation takes place in the asparagine residues, it is called as N-linked glycosylation. These O-linked and N-linked glycosylation patterns are often different in different expression systems. And I am going to actually give you one example as to how glycosylation plays a very important role in determining the biological activity of the protein. So, in addition, there could also be differences in other modifications such as phosphorylation, acetylation, sulfation and so on and so forth, and when you want all these changes to take place exactly as it happens in mammalian cells, then you have to choose for a mammalian expression system to make your recombinant protein. Now, I am going to give you one example just to tell you how glycosylation affects the biological activity of a protein. I am going to give you one example, a protein called erythropoietin, which is a very important protein. It is a glycoprotein that serves as the primary regulator of red blood cell production in mammalian cells. So, whenever the red blood cell counts in our blood goes down, it is sensed by the kidney, and the kidney now starts making a hormone called erythropoietin, and this erythropoietin now goes and stimulates red cell production in the bone marrow. So, the circulating levels of erythropoietin play a very important role in determining the hematocrit or the erythrocyte counts in your blood. So, the erythropoietin stimulates bone marrow stem cells to differentiate into red blood cells and controls hemoglobin synthesis and red blood cell concentration in the blood. Now, what is erythropoietin? It is about 30 to 31 kilodalton protein, because it is about 30,400 Dalton molecule, contains about 165 amino acids and 4 carbohydrate chains that incorporate sialic acid residues. There are several forms of erythropoietin designated by Greek letters, and these different forms of erythropoietin differ only in their glycosylation pattern or the carbohydrate content. In the infants, erythropoietin is produced mostly in liver, but the kidneys become the primary site of erythropoietin synthesis in the neonates and in the adult life. Erythropoietin production is stimulated by reduced oxygen content in the arterial blood in the kidneys. So, when you do not have sufficient erythrocytes, then your oxygenation in the blood becomes less, and the reduced oxygen content in the arterial blood is sensed by the kidney, and now the erythropoietin gene in the kidney start getting expressed, and you get erythropoietin. Now, erythropoietin, which is synthesized by the kidneys, now is released in the bloodstream. It now goes and binds to the receptor present on the erythroid progenitor cells in the bone marrow stem cells and stimulates the converts them into red blood cells. The human erythropoietin was first isolated and later purified from the urine in the 1970s. But once the recombinant DNA technology became available, the gene for erythropoietin was cloned, and several groups, especially a company called Amgen in the United States, actually devised a method to actually produce the recombinant erythropoietin by using various expression systems. So, the erythropoietin today is available in a wide different generic names. A number of companies actually produce the erythropoietin under various trade names and it became a very important recombinant protein that has tremendous use for treating anemia as well as cancer. Now, people actually found out when the erythropoietin was produced by two different expression systems, they actually found out their glycosylation pattern. These multiple bands actually show the different glycosylations of the erythropoietin that is actually produced in the mammalian expression systems, and the sialic acid content of these two erythropoietins, which are produced in two different cell lines, is more or less the same. But however, when they look at the biological activity of these two erythropoietins, one was at least twofold more active than the other. To find out what could be the cause, because the amino acid sequence is the same, the folding is the same, the glycosylation pattern seems to be the same, but still the biological activity was very different. So, when they actually looked at the carbohydrate structures which is present in these two forms, they actually found out that the form which has lower activity and the form which has higher activity, the glycosylation patterns are very different. And you can see these peaks which designate the branched carbohydrate chains are quite different in these two forms. So, the kind of carbohydrate structures in the erythropoietin can play a very important role in determining the biological activity, and with these kinds of differences it is very important you actually produce the erythropoietin in the same expression system as the happen the native, and the closest that comes to is a mammalian expression system. So, producing this erythropoietin will in yeast or insect expression systems will have a different glycosylation pattern, and therefore their biological activity will be pretty different. So, let us now spend some time to understand how you can produce proteins like erythropoietin in a mammalian expression system. And as is true for any other expression system, the first step that is required if you want to express a gene in a expression system is you have to develop a expression vector. So, let us now try to understand how do you design and develop a mammalian expression vector. Now, in this lecture today we are going to focus primarily about non-viral vectors. Okay. You can make expression vectors for expressing genes in mammalian cells using either non-viral vectors or viral vectors. Now, as you know, a number of viruses infect us all the time, and many times you can actually take these viruses, manipulate these genomes in such a way that you can actually introduce a gene of interest from the viral genome, and then take this viral genome and make a recombinant virus, and these recombinant viruses can be made to infect mammalian cells, and your gene can be expressed in large amounts and a protein of your interest can be made in huge amounts. We are going to discuss about this kind of a viral vectors or recombinant viruses for expressing recombinant proteins at a later stage in this lecture series, especially when you are going to talk about gene therapy. Today we are going to talk about how you can express genes in mammalian cell lines using non-viral mammalian expression vectors. Now, as is true for the yeast expression vector or an E. coli expression vector or an insect expression vector, in the case of the mammalian expression vectors also they need to have certain minimal characteristic features. For example, they should contain a efficient promoter elements for high level transcription initiation. They should contain messenger RNA processing signals. They should contain selectable markers, because you have to distinguish between cells which have taken up the vector and cells which have not taken up the vector. They should have plasmid sequences for propagation of these vectors in bacterial hosts, because first you have to clone your gene in a mammalian vector, put them in E. coli cells, make this vector in large amounts, then only you can go back and put them in a mammalian vector for expression. So, you need to have plasmid sequences for propagation in the bacterial hosts. The mammalian expression vector also should contain a eukaryotic origin of expression, eukaryotic origin of replication usually from an animal virus such as SV40 virus, so that it can also replicate in mammalian cells. The origin of replication from E. coli is a must for all the mammalian expression vectors, because once you clone your gene into the vector, you have to make this plasmid in large amounts in bacterial cells. So, these plasmids have to multiply in E. coli cells, therefore, you should have an origin of replication that functions from E. coli. You should also have promoter sequences that drive not only your cloned gene, but also the selectable marker genes. So, we will discuss about what kind of selectable markers are actually used for expression of genes in mammalian cells. Of course, you should also have multiple cloning sites for cloning your genes, as well as transcription termination sequences, as well as polyrelation signals from viral or mammalian origin, so that from the promoter the gene is expressed and the transcription is properly terminated, and then your messenger RNA is polyrylated, and these polyrylated messengers can then be translated by the eukaryotic transcription machinery or translation machinery. In addition, many of the modern mammalian expression vector contain what are called as matrix attachment regions, as well as chromatin insulators and locus control regions. We will discuss these new features of mammalian expression vectors at a later stage in the lecture series. Now, viral genetic elements have been extensively used to construct a number of eukaryotic expression vectors. Now, viruses are highly efficient replicators, and viral gene expression is adapted to eukaryotic systems. As I told, the animals as well as humans are often bombarded by a number of viruses. We are infected by a number of viruses. They have natural tropism to infect select cell types in our body. So, these viruses, when they enter the body, they actually use the host transcription machinery to replicate their to transcribe their RNA, and therefore they contain very powerful promoters to which the host transcription factors go and bind and activate the transcription. So, if you can choose some of these promoters in these viral genomes which control the expression of the viral genes, and you can take these promoters and design eukaryotic expression vectors, so that you can express your gene of interest in very high amounts. So, some of the very strong viral promoters which are very routinely used in expressing genes in eukaryotic cells are the cytomegalovirus immediate early promoter, which is one of the most widely used mammalian expression vectors, contain the same promoter, and usually these mammalian expression vectors also contain a intron that is derived from the cytomegalovirus promoter, cytomegalovirus genome. If you remember some of the early lectures that I gave in this lecture series, the splicing, when the messenger RNA is synthesized in eukaryotic cells, as the transcription proceeds, some of the post transcriptional modifications such as splicing, polyadenylation, they all occur simultaneously, and all these processes are coupled. And there have been many instances where when you have an intron, these RNAs are spliced efficiently and they are also polyadenylated efficiently. And if the RNAs which are not spliced are not efficiently polyadenylated, and therefore if you want a efficiently transcribed gene and if you want a good amounts of polyadenylated RNA to be made, it is better to include an intron in the vector, so that the RNA that is synthesized from these vectors are spliced and therefore they are also efficiently polyadenylated, because splicing and polyadenylation is linked. So most of the eukaryotic expression vectors also contain an intron, so that immediately after transcription the RNA is spliced and such RNAs are likely to be polyadenylated more efficiently. And the promoter regions should contain regulatory elements, and if you want your gene to be constitutively expressed all the time, you include constitutive promoters, and if you want to be regulated, then you have to include a regulatable or a inducible promoter depending upon the choice. So if you want to express your gene constitutively, mostly use viral promoters, and not only use viral promoters, you also use enhancers, polyadenylation signals, introns, replication RGs, and there are what are called as internal ribosome entry sites or IRES elements. Most of these actually are incorporated from the animal viruses or human viruses, because these are very well characterized. So the eukaryotic promoters which are commonly used in mammalian expression vectors are as I said, the cytomegalovirus promoter is one of the most extensively used and very highly expressed very powerful promoters that are routinely used in mammalian expression vectors. You also use the Simian virus 40 promoter and enhancer, or sometimes you can use vaccinovirus promoters. You can also use what are called as viral long terminal repeats or LTRs, which contain very powerful promoters. For example, the mouse mammary tumor virus contains a long terminal repeat which actually contains the glucocorticoid response elements, and therefore when you use mammalian MMTV LTRs for in your expression vectors, such in vectors the expression of your transgene can be regulated by glucocorticoids. So you can turn on the expression by adding glucocorticoids to the culture medium, and you can turn off the gene by removing the glucocorticoids in the medium. But if you want a constitutive expression, you can use the Rous sarcoma virus long terminal repeat or the HIV LTR. So depending upon whether you want to express the gene constitutively or in an inducible manner, you can use different viral regulatory elements. In addition to these promoters and enhancer sequences from the animal viruses, you can also use the promoters and enhancer sequences from a number of mammalian genes which have been very well characterized. For example, you can use actin or glyceraldehyde 3-phosphate dehydrogenase promoters. As you know, actin is expressed in almost all cell types and is one of the most abundant proteins in the cell, so it is a very powerful promoter. Similarly, GAPDH, again because of its housekeeping functions, is abundantly expressed in a number of cell types. So if you want to express your gene constitutively in a number of cell types, you can use either actin or a GAPDH as your promoters of your choice. You can also use promoters of genes which are expressed in a tissue specific manner. For example, if you use an albumin promoter, you can express your transgene only in liver cells or a liver cell lines. You can use, for example, promoters like neuron specific kinolase, which is a very powerful promoter, and when you use this promoter, you can express your genes only in neuronal cells or neuronal cell lines. Similarly, you can use mammalian promoters which are inducible, for example, metallothionein. You can use metallothionein promoter. In such cases, you can induce the expression of your transgene by adding metals into the medium, into the culture medium, or you can also clone your gene substream of promoters containing steroid hormone response elements. One example I gave you is the mouse mammary tumor virus, which contains the glucocorticoid response elements, and in such cases the expression of your gene can be induced by adding glucocorticoids. So a wide variety of promoters are available, either of animal virus origin or of various mammalian origin, and you can use any one of these promoters and design a expression vector of your choice. So a typical mammalian expression vector contains a strong promoter, and as I said, this strong promoter can be either of an animal viral origin or it can be of a mammalian promoter, and usually you include an intron immediately downstream of the promoter sequence, and then you contain a multiple cloning site into which you introduce your transgene, and the transgene should contain a ATG codon, so that it is with actually a Kozak consensus sequence, so that it is properly translated efficiently. Should also have a proper stop codon at the end, and it should also have 3' untranslated sequences, so that such as the AAUAAA or the polyadenylation sequences, so that the RNA is transcription stops and the RNA is properly polyrylated. And many a times when you want to purify your protein that is made from your construct, a very simple using very simple methods, you can actually include certain tags. For example, you can use a purification tags, such as if you include 6 histidines in the amino terminal region of the or the carbox terminal region of your gene, then you can purify such recombinant proteins easily using a nickel affinity tag, because histidine binds to nickel, and using a nickel column you can easily purify recombinant proteins by virtue containing a histidine tag. And many a times you can also add tags like a MIC tag or what is called as flag epitopes. These are all very short amino acid sequences which are usually incorporated in front of recombinant proteins, and in such cases detection of your protein becomes very easy, because antibodies against these epitopes, flag epitopes or MIC epitopes, are commercially available, and therefore you do not have to actually have an antibody against your recombinant protein. By incorporating these epitopes into the amino terminus or carbox terminal region of your gene, you can express these proteins containing MIC epitopes or flag epitopes, and expression of your recombinant proteins can be easily detected using antibodies raised against these particular epitopes. So by using histidine tags you can purify your recombinant protein very easily by nickel affinity column, or by introducing epitopes like MIC epitopes and flag epitopes you can easily identify the expression of your proteins using antibodies available against these epitopes. So there are also many modern expression vectors which also contain, as I said, certain barrier elements and insulators and so on and so forth. We will discuss some of these very unique developments a little later in the lecture series. This is a typical eukaryotic expression vector I have shown here, which contains a both viral as well as bacterial sequences. The viral sequence of viral origin are shown by asterisks in red color. For example, it contains a cytomegalovirus promoter, it contains a SV40 origin of replication, so that if you now introduce this vector in mammalian cell lines which have been transformed with SV40 large T antigen, then every time the cell divides your vector also divides. So when you transfect cells and cells start dividing, the daughter cells also will contain your plasmid your vector. It also contains the SV40 polyadenylation signal, so that your RNA gets properly polyrylated. The blue diamonds actually denote the sequences derived from the bacterial origin, the ampicillin resistance gene, so that you can clone your gene first in the multiple cloning site and then make this vector in large amounts, or select these bacterial cells containing harboring the gene by ampicillin resistance marker. It also contains a PUC origin of replication, so that this plasmid can be efficiently replicated in the E. coli cells. So a typical mammalian expression vectors contains a number of sequences from animal viruses such as cytomegalovirus promoter, SV40 polyretinal signal, and so on and so forth, and it also contains several bacterial sequences for them to be introduced to bacteria and select the bacterial cells as well as for replication in bacterial cells. Usually when you want to introduce your expression vector, mammalian expression vector, into eukaryotic cells, you can do two things. You can express your transgene, or you can express your expression vector. You can induce the expression of the gene in the expression vector in a transient mode, or you can permanently integrate your gene of your interest into the genome of the mammalian cell. So when you want expression only for a short period of time, and when you do not really want your transgene to be integrated into the host chromosome, such the transfection is known as the transient transfection. So in the case of a transient transfection, standard non-selectable transformations of mammalian cells that replicate the plasmid DNA, but do not faithfully segregate the plasmid DNA to progeny in the time of the plasmid will be lost from the cultured cells. So if you just want, for example, to study certain promoter sequences, or study or express a gene only temporarily, and you do not want all the cells to be transfected, then you adopt for a transient transcription. Usually for many basic research studies. For example, when you want to do some functional studies or when you want to make some deletion mutants of certain genes and look at their functional domains, you use transient transfection studies. But usually for recombinant protein production in large scale, you go for what is called as a stable transfection. So that not only the cell, once the gene gets introduced into the mammalian cell, it gets stably incorporated into the host genome, and therefore every time the cell divides, your transgene along with the promoter also divides, and therefore the daughter cells also will contain your gene of your interest. And so in the case of a stable transfection, you have to actually select those cells which have taken up your vector, and therefore you need to have appropriate markers so that you can select those cells using that marker. So the main difference between a transient transfection and a stable transfection is, in the transient transfection the gene is not incorporated into the host genome and you do not actually select the cells for which have got stably transfected the gene of interest, whereas in the case of a stable transfection you actually introduce a selectable marker in the vector so that it can actually select those cells which have actually taken up the vector. So a selectable marker is included in the plasmid vector, so that after the cells have been transfected, the rare cell that incorporates the plasmid DNA into the genome through either DNA repair and or homologous recombination can be isolated and cloned. So the progeny of transformed cells will inherit the plasmid, including selectable marker gene such as an antibiotic resistance gene. So these are the two different methods. You can either use a transient transfection method or you can use a stable transfection method for expressing your genes in a mammalian cells. Accordingly, if you want to do a transient transfection, you have to choose a different kind of a vector. If you want to do a stable transfection, you choose a different kind of a vector. So in both the cases, the vector should contain a strong promoter. In this case, for example, the vectors contain a cytomegalovirus promoter so that the protein can be expressed constitutively in the cell line. It also contains for SV40 origin of replication, so that when you introduce this vector in a mammalian cell line which contains or which has been transformed with SV40 large T antigen, every time the cell divides, the vector also divides. They also contain a origin of replication from PBR322, so that these can be replicated efficiently in bacteria. They also have an ampicillin resistance marker, so that they can be E. coli cells which are taken up the plasmid can be selected. But the only difference between a vector for a transient expression, vector for a stable expression is that stable expression vector actually contain say a selectable marker gene. In this case, the aminoglycoside phosphotransferase 2 gene, often referred to as the neo-R gene or a neomycin resistance gene. And you can see the neomycin resistance gene, which is under the control of a eukaryotic promoter, is present in a vector meant for a stable expression, whereas a neomycin selection marker is absent in a vector which is meant for a stable expression. Now let us spend some time to understand how do you actually select the eukaryotic mammalian cells which are taken up the plasmid DNA or the vector DNA. The very commonly used selection marker for making stable cell lines expressing your gene of interest is the neomycin resistance gene, and you actually select the cells by adding a compound called G418. Now G418 is actually used for the selection and maintenance of eukaryotic cells which express the neomycin resistance gene. Now what is G418? G418 is an aminoglycoside antibiotic produced by Micromonospora rhodorengia. The G418 blocks polypeptide synthesis in eukaryotic cells by binding irreversibly to the ATS ribosomes and therefore disrupting their proofreading capability. So this is the mechanism by which the neomycin resistant genes offer. So how do you develop resistance to G418? The resistance to G418 is conferred by the neo-R gene from the transposon TN5 encoding an aminoglycoside 3 phosphotransferase. So this protein inactivates G418 by covalently modifying its amino or hydroxy functions, therefore inhibiting the antibiotic ribosome interaction. So those cells which express the neo-resistant gene modify the antibiotic so that the translation is not blocked, whereas those cells which are not taken up the neo-resistant gene, when you add this antibiotic, it goes and blocks the translation and therefore these cells die. So cells which have taken up your plasmid become resistant to G418, cells which are not taken up your vector become sensitive and therefore they die. Many a times the exact concentration of G418 that you have to use to select mammalian cells which have taken up these vectors often varies from cell line to cell line. The optimal concentrations of G418 required to kill a specific host cell line has to be determined by treating the cells with several concentrations ranging from 100 microgram per ml to 1 milligram per ml, and after the treatment the cell death occurs rapidly, allowing the selection of the transfected cells with vectors carrying the neomycin resist genes within about a week's time. So, if you once you do the transfection with your stable vector, you add G418 and allow the cells observe the cells for about a week time. All those cells which are not taken up the G418 they all die, whereas cells which have taken up the vector become resistant to G418 and you get what are called as distant colonies, and these colonies can be cloned and then be used for further analysis. So, different cell lines have required different amounts of G418. Some cells require less G418 to be killed and some cells require more G418 to be killed, and this has to be standardized depending upon what kind of cell line you are using for expressing your gene. So, the two major distinctions between a transient transfection and a stable transfection is the vectors which are used for transient transfections do not contain the selection markers such as neomycin resistance gene. So, when you introduce this transient transfection, the gene may not be introduced in all the cells, only some genes take up the genes and these genes remain extra chromosomal and the DNA does not you are not actually selecting for those genes in which the DNA has got stably incorporated. So, they remain extra chromosomal and therefore, they replicate, and some cells may not take up the plasmid, but you are not selecting for those cells which are not taken up the plasmid. In the case of stable transfection, following transfection of your vector, you actually add antibiotics such as G418 and all those cells which have taken up your gene and have got stably integrated this gene, every time these cells divide, your gene of interest also divide, and therefore, those cells get selected, they become resistant for the G418 and therefore, ultimately you end up with colonies in which your transgene has got stably integrated into the host chromosome, because not only their progeny also become drug resistant. So, in addition to G418, other very popular selectable markers which are used for selecting vectors following transfection to mammalian cells are what is called as methotrexate is another drug that is actually used, and the selection marker which is used is called as the dihydrofolate reductase gene, and by those cells which express dihydrofolate reductase become resistant to methotrexate, therefore you can select these cells. There also you can use hygromycin Z phosphotransferase gene as a selectable marker, and you can actually select those cells using hygromycin B, which is again an inhibitor of protein synthesis. You can have thymidine kinase. Again you can select those cells which express thymidine kinase using aminopterin, which actually inhibits de novo purine and thymidylate synthesis. You can use XGPRT or the xanthine guanine phosphoribosyl transferase. Again the compound you can use to select cells or mycophenolic acid, which inhibits de novo GMP synthesis. You can also use adenosine deaminase as a selection marker. In this case you can use a modified nucleotide for selecting those cells which are resistant to this, which express this particular gene. So, a number of selectable markers can be used for selecting, for developing or for isolation of stably transfected mammalian cells which are expressing your gene of your interest. I am going to just spend some time to express about how you select for methotrexate resistant colonies for expressing, because this is one of the markers which are very efficiently used for recombinant protein production in a cells called Chinese hamster ovary cells. So, methotrexate actually inhibits the enzyme dihydrofolate reductase. The dihydrofolate reductase host cell with the DHFR gene is present on the cloning vector and is therefore linked to the target gene. And what you do is, once you do the transfection with your stable expression vector containing your gene of interest, you slowly start expose the cells to increasing concentration of methotrexate, and when you do that, the cells, some of the cells which contain this DHFR gene, the DHFR gene gets amplified to cope up with the increasing concentration of methotrexate, and therefore, by increasing with different generations, by slowly increasing methotrexate concentration, you can increase the copy number of your gene, and therefore, more the copy number, they can tolerate higher concentrations of methotrexate. Therefore, by gradually increasing methotrexate concentration, you can finally select a cell line which contains very high copy number of your transgene and therefore expressing very high levels of your protein. This is the brief how you do the selection protocol. So, you do the transfection in the absence of the inhibitor, and usually most of the cells will contain one copy of your vector, very rarely you will have two copies, and then you start selecting those cells in the presence of the inhibitor. So, those cells which are not taken up the vector, they all die, they become sensitive to methotrexate, but there will be some cells, as you start increasing the methotrexate concentration after every generation, slowly in some of the cells the DHFR gene gets amplified, that is shown in the indicator. For example, in this case many cells contain only 2 copies, but some cells may contain amplify the DHFR gene, so they contain 4 copies, or if you express now higher concentration, they may now increase to 6 copies, 8 copies, 10 copies, and so on and so forth. So by gradually exposing the cells to higher concentrations of methotrexate, you can actually score for cells which are tolerant to higher concentrations, because the gene got amplified and therefore they have more and more of DHFR protein present, and accordingly your transgene copy number also goes up, therefore the expression level of your protein of interest also goes up. So this is in brief how you express how you design an expression vector, mammalian expression vector, and using either viral enhancers or viral regulatory elements or mammalian promoter elements. And once you design, you can either design a transient expression vector or a stable expression vector, and then you select in the case of stable transfection using compounds like G418 or methotrexate, you can actually select for stable transformants expressing high levels of your protein of your interest. I am going to now spend some time to express another very important feature of new kind of expression vector called as bisystonic expression vectors. Now bisystonic expression, so far we talked about expression vectors in which you are introducing one gene and therefore only one protein can be made from the vector. Now there are also what are called as bisystonic expression vector, and they incorporate a very unique feature, what is called as a internal ribosome entry sites. Now let us now discuss what these are. Bisystonic expression vectors actually contain what is called as an internal ribosome entry site, abbreviated as IRAS element, and this is usually derived from the encephalomyocarditis virus or the EMCV for translation of open reading frames. As you all know, for the normal translation of the eukaryotic messenger RNAs, you require cap, the 5' cap, because the ribosome translation machinery actually recognize the ribosome cap and then only the 2 subunits assemble. So for the efficient translation of eukaryotic messenger RNAs, you require a capped transcript, or a 5' cap is very, very important. But there are certain viruses whose messenger RNAs can be translated even in the absence of this cap structure, and it turned out these viruses contain what is called as a internal ribosome entry site. This is actually a DNA sequence, and when you have these DNA sequences, ribosome can still bind to these IRAS elements and do not actually require a 5' cap. So many of these viruses have developed a mechanism by which they actually inhibit the capping mechanism, and therefore the host messenger RNAs will not be translated efficiently, but their own messenger RNAs, by virtue of containing these IRAS or internal ribosome entry sites, can be efficiently translated. So they make use of the host translation machinery for the translation of their messenger RNAs rather than translation of the host messenger RNAs. So people have actually taken advantage of these internal ribosome entry elements and incorporate these internal ribosome entry elements in a expression vector. For example, here you have an expression vector where you have 2 multiple cloning sites, and the first multiple cloning site is downstream of the cytomegalovirus promoter. So when you clone your gene of your interest here, the RNA polymerase, host RNA polymerase binds to promoter and transcribes your RNA. But you also have an internal ribosome entry site, and you can clone your second gene into the multiple cloning site which is present here. Therefore 2 genes can be cloned, one before the IRAS and one after the IRAS. So a single messenger RNA which is transcribed from this expression vector now gets translated in a cap independent fashion. The first gene is translated in a cap dependent fashion, where the second gene is translated in a cap independent fashion, because ribosomes can bind to both the cap structure, they also can bind to the internal ribosome entry site. So simultaneously 2 proteins can be synthesized from a single messenger RNA. That is why they are called as bisystonic vectors. So the bisystonic vectors are designed to drive the transcription of a bisystonic message under the control of a strong viral promoter such as the same promoter, and these vectors permit co-expression of 2 genes of interest. Genes cloned into MCS1 are expressed in a cap dependent manner, and genes cloned in the subsequent MCS are translated in a cap independent fashion under the control of the internal ribosome entry site. So you can make 2 proteins from a single vector. For example, in this vector you can clone your gene of your interest downstream of the cytomegalovirus promoter using this MCS, and you also have another gene coding for the green fluorescent protein, which is a very good reporter gene and which is under the control of an IRA sector. So when you clone your gene of your interest in the MCS and when you transfer this vector into a mammalian cell line, both the proteins will be synthesized, and you can usually score genes which got transfected by scoring for the expression of a GFP. By just looking under a UV light, you can see which kind of cells have taken up your plasmid, what are the cells which are expressing. So, the internal ribosome entry site from the encephalomyocardite virus is a non-coding RNA fragment that can initiate high levels of cap independent protein synthesis in mammalian cells as well as in cell-free extracts. The IRS is localized to a genome fragment of about 430 bases, immediately 5' to the translation initiation codon or the AUG, which begins the viral polyprotein open reading frame. When this region was excised and linked to other portion of the virus ORF, the resulting transcripts were translated even in the absence of 5' capping. This discovery that these animal viruses such as the encephalomyocardite virus as well as polio viruses, they contain this kind of internal ribosome entry sites, this discovery was actually commercialized in 1990 by a company called Novagen to form the first bisystemic vector called as P-Site1. So, site caps transfer cap independent translation enhancer. So, you do not require capping anymore, and therefore, you can use these vectors to generate RNAs in a translate RNAs in a cap independent fashion. Following this cap independent translation enhancer was later named as internal ribosome entry site, and a company called CloneTech developed a number of bisystonic vectors which contain these internal ribosome entry sites. One of the advantages of using these kinds of bisystonic vectors is that suppose you want to express a protein which is a heterodimer. It contains two different subunits. So, what you can do is you can clone this two genes coding for each subunit, one under the control of the promoter, another downstream of the IRES, and by just transfecting one vector, both the subunits can be made in a single cell and they can heterodimerize and you can get a functional protein. So, when you want to express many proteins which are heterodimers, you can express both subunits using these kind of bisystonic vectors and you can get a functional protein by using a single vector rather than cloning each one of the subunit genes into two different vectors and co-transfecting these plasmids. So far we have discussed about what kind of vectors are people use for expressing genes in mammalian cells. Now, let us now try to understand what kind of a transfection methods one has to employ if we have to introduce this gene into vector into the mammalian cells. Now, as you are aware, many of these vectors that we are discussing, they are all negatively charged. The mammalian cells have a bilayer which is also negatively charged. Therefore, mammalian cells cannot take up the DNA just like that. So, if you have to introduce your vector into mammalian cells, you need to evolve some specific mechanisms or specific treatments. So, some of the methods that are routinely used for introducing these vectors, expression vectors in mammalian cells, are by doing what is called a calcium phosphate precipitation. What you do is you take your DNA which is negatively charged and mix the DNA with calcium salts, usually calcium chloride or calcium chloride. So, the calcium which is negatively charged or which is positively charged binds to the negative phosphate groups of the DNA and therefore makes it kind of neutral, and therefore, these calcium phosphate crystals are now efficiently taken up by the mammalian cells. So, you are temporarily neutralizing the negative charge of the DNA and therefore, the DNA can be taken up by the plasmid membrane inside. This is a very old method of transfection which has been very successfully used for number of basic research applications for understanding eukaryotic gene expression in the 90s and early 2000, but many modern methods of transfection have now taken over. Very rarely now people use calcium phosphate mediated transfection these days. The most preferred method of introducing vectors, expression vectors into mammalian cells is now using what are called as liposomes or cationic lipids as well as by electroporation. So, in the case of liposomes, the vector of your choice is mixed with lipids to form what are called liposomes, which are nothing but small vesicles with the DNA inside, and these DNA bearing liposomes fuse the cell membrane and therefore, the DNA gets inside the cell. This is now shown pictorially here. So, you actually vortex your DNA of interest with what are called as the lipid solutions. As you know, the lipid has both the hydrophobic and hydrophilic positions. So, they form actually lipid vesicles and your DNA gets trapped inside these lipid vesicles, and if you now add this into the cells in culture, the lipid actually fuses with the lipid bilayer and the DNA gets delivered inside. So, it is a very popular method of transfecting or mammalian cells by using expression vectors. These days you also have a variation of these liposomes. You also have what are called as cationic lipids, which are nothing but positively charged lipids, and therefore, you can use these positively charged lipids. When you mix your DNA with these positively charged lipids, the positively charged head of the cationic lipid now binds to the DNA and they form a neutral lipid particle which is now efficiently taken up by the mammalian cells. In the case of so this is what happens, the liposomes adhere to the cell membrane and the lipids fuse with the cell membrane and the DNA gets inside, and therefore, the DNA goes inside the nucleus and the DNA of interest gets transcribed. This is how cationic lipids and liposomes can be used for introducing your genes. In the case of electroporation, you actually take your gene of interest, mix it with the cell of your choice, and then subject them to a short electric current, and when you give this, apply a specific voltage, it creates pores inside the membrane cell membrane, and DNA gets inside the mammalian cells. So when you apply an electric pulse, when the cells containing DNA, the pores are made inside the cell membrane, the DNA gets inside, and therefore, and once the DNA gets inside, you can then either, if the vector contains a selection marker, you can apply the selection pressure and select for stably transfected cells, or you can just go ahead with the transient transfection. So three very popular methods of introducing mammalian vectors into mammalian cells is calcium phosphate precipitation, electroporation, as well as lipid mediated gene transfer using either liposomes or cationic lipid mediated gene transfer. So some of the very important factors which affect the expression of your genes in mammalian cells are what kind of a vector you design, as I said, depending upon the promoter, as well as other sequences like inclusion of intron, proper polynation signals, and so on and so forth. Efficient mammalian expression vectors have not been designed. So designing an appropriate highly efficient mammalian expression vector is one of the major requisites for efficient expression of your transgene or producing recombinant proteins in a large amounts. The next important step is what kind of host cells you are choosing. There are many host cells which cannot be efficiently transfected, and there are also many host cells which do not actually carry out appropriate post-translation modifications. Therefore, choosing the appropriate host cells is very, very important once you design an appropriate expression vector. Then the most important another important criterion is the growth conditions. So what kind of growth conditions you can actually grow these host cells into high cell densities so that you can produce your protein in large amounts. So a number of parameters actually determine whether you can produce a recombinant protein in high levels or not. So as I said, if you want to do transient transfection, usually people choose something like the CAS cells or the bovine hamster kidney cells or human embryonic kidney 293 cells. These are the usual mammalian cells people choose for doing transient transfection studies, or you want to express your gene in a transiently. Whereas if you want to express your gene in a stable form, usually the Chinese hamster ovary cells are mostly preferred for stably transforming genes and especially for producing recombinant DNA proteins in large amounts. For example, erythropoietin is primarily produced using the Chinese hamster ovary cells. So the Chinese hamster ovary cells are the most widely used cells for mammalian expression of mammalian genes, especially for large scale recombinant protein production, and the CHO cells provide very stable and accurate glycosylation. They offer post translation modified product that is more or less that is almost the same as the natural protein. In fact, the CHO cells were introduced in the early 1960s as a viable epithelial cell line containing 2 female X chromosomes. So the CHO cells, or as it is called, is very widely used for recombinant protein production, and the erythropoietin, which we discussed in the beginning of this lecture, is actually produced using the CHO cells. Several mammalian expression vectors and cell lines are now commercially available. For example, in vitro gen cells, actually what is called as a Freestyle 293 expression system. If you just buy a kit, they supply the vector, they contain sublines and all the transfection reagents, as well as for methods for stably selecting the stable cell lines, and you can also the growth conditions, and all this can be you don't have to slog. You can simply buy these kits and you can express your gene of your interest. For example, using this kind of a Freestyle 293 system, it has been shown that a number of proteins can be expressed at very high levels, from grams per liter to even milligrams per liter. A very high level of expression can be obtained using these kinds of commercially available vectors and commercially available cell lines. Here, for example, using this particular vector and using the cell lines, in vitro gen actually tells you that number of proteins like erythropoietin, factor 9, immunoglobulins have been expressed at milligram levels using mammalian cell lines. There are actually companies such as G0 where you don't have to learn how to express proteins. If you simply tell them what kind of a mammalian gene you want to be expressed and pay the money, they will design the appropriate expression system, they will make the expression vectors, they will introduce into the cell lines, and they will express the protein and give you the recombinant protein of your choice. So these are called the custom protein expression systems are now available. You can go to these companies and just pay the money, and they will express the protein of your interest and give you the protein. So let's now spend about some time to understand cell-free protein synthesis. So far all our focus has been on expressing genes inside the cells, whether it is E. coli cells, whether it is yeast cells or insect cells or mammalian cells. But there are also now cell-free systems which are available, so you can make your protein in a test tube without any cells in a cell-free manner. Now, the first recombinant protein expression in a cell-free system dates back to 1961, wherein Nirenberg and Mathai actually demonstrated if you have an RNA which contains U residues, you can actually make a polypeptide containing phenylalanine. This is one of the first successful demonstration of a cell-free protein synthesis. In fact, it made a very important contribution for the understanding of the genetic code, and they got the Nobel Prize for it. Following it, people started making this kind of cell-free extracts. For example, cell-free extracts have been made. So, a cell-free extracts actually should contain what? It should contain all the amino acids, it should contain all the amino acid tRNAs, it should contain the ribosomes, and therefore, if you have and also have a proper energy source, therefore, if you simply add an RNA, the ribosome can translate to your RNA. So, cell-free extracts have been made. Cell-free extracts capable of protein synthesis have been made from E. coli, they have been made from rabbit reticulocyte lysates, and they have been made from wheat germ. What is the advantage of a cell-free protein expression system? You can actually use the cell-free protein systems to make proteins which are actually toxic to cells. You can use proteins which can be made, soluble proteins can be made using cell-free systems which normally go into inclusion bodies in E. coli. Proteins which are sensitive to intracellular proteases can be now made using cell-free protein systems, and if you want to label proteins for with certain heavy isotopes for NMR studies, cell-free systems can be made use of. So, the cell-free protein expression system is a viable alternative for making proteins inside the cells. As I said, the E. coli cell-free protein systems are now commercially available. It has a very simple translation operators and a comparatively simple control of initiation. Following this, the rabbit reticulocyte lysate systems have been developed for efficient in vitro protein synthesis system. Reticulocytes are nothing but immature red blood cells specialized for hemoglobin synthesis, lacking nuclei, but they have a complete translation machinery. So, what you do is you take these reticulocytes and degrade the endogenous globin messenger RNA present in the reticulocytes by treating them with micrococcal nuclease which chews up all the RNA, and then you inactivate the nucleus by chelating with EGTA, and then exogenous proteins are synthesized at a rate close to that observed in the intact reticulocytes by adding messenger RNAs into this reticulocyte lysate. So, both capped as well as uncapped RNAs can be translated using this kind of a rabbit reticulocyte lysate systems, and you can get nearly full length proteins can be synthesized using the rabbit reticulocyte lysates. The same way, using a plant source, you can actually make a wheat germ lysate. Again, using the similar procedure, you destroy the endogenous RNA present in the lysate by micrococcal nucleus, and then you add your messenger RNA. Your messenger RNA will get translated, and your protein can be made. As I said, it has an advantage. Many proteins which are toxic to cells can be expressed using these in vitro protein expression systems. They can be used for a number of structural studies for like crystallography, mass spec, NMR, and so on and so forth. It eliminates the cost and time required for cell-based systems. You do not have to maintain the cell lines, you do not have to optimize the expression, you do not have to do transformation or transfection. You have a nice cell-free system for making your proteins. A number of commercial extracts are available, for example, Promega cells, all these E. coli cell-free system, rabbit reticulocyte lysates, as well as wheat germ extracts. You can simply buy these cell-free systems and simply put your RNA of interest there, and you can get your protein of interest. Two important breakthroughs occurred in the cell-free expression systems for mammalian systems. One is called as a coupled transcription translation systems, and what is called as a continuous exchange cell-free system, or the CECF system. Now, in the early in cell-free systems, you have to first transcribe your gene using certain prokaryotic RNA polymerases like T7 RNA polymerase, 3,7 RNA polymerases, and then take this RNA, purify this RNA, and then add this RNA to a cell-free translation system to make your protein. Now, in the case of the coupled transcription translation systems, you simply add your plasmid which contains your gene. In the same system, the gene will get transcribed by the phage RNA polymerase, and then the RNA will get translated. So, in one shot, both transcription and translation takes place in the same system, and therefore, it is called as a coupled transcription translation system. Whereas, in the case of the continuous exchange cell-free system, it is a very important breakthrough that happened, because in the normal cell-free systems, one of the important problems is you cannot make large amounts of your protein because the energy gets depleted. Whereas, in the case of continuous exchange cell-free systems, energy is continuously replenished, therefore, a large amount of protein can be made, and even milligram amounts of proteins can be made using this kind of a cell-free translation systems now. So, I have just taken some of the protocols which are given on the company websites like Promega actually gives you how actually you can do both transcription and translation in the using this coupled transcription translation systems, and you just have to buy these kits and you can make your protein of your interest. Similarly, using the commercial kits like Expressway, you can actually synthesize large amounts of these kinds of proteins using this kind of coupled transcription translation systems, and again the company gives the exact protocols how to go about and do these cell-free transcription machineries. Roche again sells very efficient cell-free transcription translation systems for making milligram amounts of proteins, and one can go to the Roche website and get the mechanism by which you can actually make these proteins. So in this chart I have actually listed all the expression systems we have discussed so far. We have discussed about E. coli expression systems, we discussed yeast expression systems, insect expression systems, and mammalian expression systems. And again you have to choose your rationale and your decision to see what kind of expression system you have to choose. Each one has its own advantages and disadvantages. Ultimately the choice depends on the particular gene of your interest you are interested in, what kind of protein you are interested in, and if you have to make a protein which if you have to make a protein which does not require post translational modification, you go for an E. coli system and then gradually try out other systems, and ultimately if none of the systems work, ultimately you choose a mammalian expression system because that is the most expensive. So with this we have come to the end of this lecture. So we have discussed various aspects of non-viral methods of introducing genes and making proteins in mammalian expression systems. What we will do in the subsequent class is to look at some of the viral based vectors for gene expression in mammalian cells. What kind of viruses are people using to introduce genes in mammalian cells, and how various viruses such as adenovirus, retroviruses, polioviruses, lentiviruses, and vaccinia viruses, these viruses can be modified so that genes can be introduced into those genomes and recombinant viruses can be generated, and these recombinant viruses can now be infected to various mammalian cells, and proteins can be expressed in very efficiently in large amounts. I think I will stop here.